Hello and welcome to the unit Vertebrate Biodiversity. I am the unit coordinator for Vertebrate Biodiversity. My name is Dr. Christopher Turbill. There's my email address. Please email me if you wish to have a chat about the unit, or you can call me. There's my phone number, my office phone number, extension 1456, and there's a picture of what I look like. I can't be with you in person this week because I need to be in the field helping my PhD student in the Northern Territory at ghost bats. So sorry about that. Instead of pre-recorded the lecture for you to listen to right now, next week I'm back on campus and I will meet some of you during the prac class, the first prac class on Tuesday. If you're in stream A, you will have your first prac on Tuesday, either in the morning or afternoon. This Tuesday it is, in the second week. If you're in stream B, then you'll have the same prac in the third week on Tuesday. On Thursday of week 2, I will present the second lecture in person and I will see you then. So there's a vertebrate animal. The vertebrates include those animals that we're most familiar with. They're often large and very active. They have a distinct head, carrying in almost all groups jaws and other well-developed sensory organs like eyes and chemical sensing nostrils. And again, in most groups, 2 pairs of appendages sporting either fins, arms or legs. Along the body are large segmented muscles, and the body is supported by an internal skeleton centered around a vertebral column which prevents compression of the body. These sensory organs collect information and deliver this information via a nervous system to a relatively large brain in the head. And this sensory system allows the animal to be very active, such as this very effective predator, of course. The activity in large brains of vertebrates require an efficient method to extract oxygen from the environment, from either water or air, via gills or lungs respectively. And they also require a closed and high-rate delivery system, a vascular system, to provide that oxygen to where it's needed in the cells of the muscles in the brain. And the high rate of metabolism of vertebrates requires a well-developed extracellular digestive system, of course, to extract the nutrients that will burn with that oxygen to deliver to make the ATP that all those active cells and organs need. Really, there's no doubt about it that although most animal species are invertebrates, of course, they're much more diverse. There's many more invertebrate species than vertebrates. The vertebrate animals are nevertheless some of the most spectacular and awe-inspiring and familiar of all animals. So what's this unit about? First of all, we will define what vertebrate animals are, we'll look at where they've come from, and really the unit follows different groups of vertebrates and looks at the main organ systems and asks how have they evolved to solve the challenge of living in different environments over time as these organisms have evolved over time by natural selection. What are the key adaptations of vertebrates that enable them to be successful in the face of different environmental challenges, like I just said? So this unit really takes the perspective of surviving in different environments. And what have the animals evolved that helps them to solve those challenges that the different environments have provided? We'll also look at the diversity of vertebrate animals, especially from an Australian perspective, and we will look at what adaptations have been critical in Australian environments in particular. Vertebrate animals also provide functions, ecological functions to ecosystems. We'll look at what some of those functions are, why they're important. And we'll also, of course, examine how human activity is threatening vertebrate animals with extinction. Finally, a good chunk of the unit focuses on how to conduct scientific studies to understand the relationships between environmental conditions and resources and the distribution and abundance of vertebrate animal groups. We'll do that via you will do that by conducting a scientific study in small groups to understand the factors that affect the abundance and distribution of bird species. It's one of the assessments that the unit focuses on. Alright, so here is the unit schedule over the semester. This is the first lecture we're having now, of course. The second lecture, I'll see you in person, will be about living in water, fish and sharks. And here are the prac class schedule. We've had to change the practice classes this year to a Stream A, Stream B type of schedule because there's quite a few students in the unit. So there's been a bit of a stress at the last minute getting the allocate system and that sorted out, so I apologise about that, but hopefully it should be sorted by now. Basically, the students will be, you will be in either Stream A or Stream B, and so each practice is repeated, of course, for each stream of students, week by week. And then on the Tuesday when the practice occur, the practice are repeated as well, of course, either in the morning or afternoon. The other thing to note about the schedule is the associated with each week are chapters in the textbook Poe, which I'll get to in a moment. And I've noted down which chapters you should read as background material for that part of the unit, for that lecture. I really do recommend that you read those chapters. Gives you essential background information, really helps you understand the unit a lot more than just listening to my lectures. Get to what I think you should be doing in regard to the unit, a bit later as well. The schedule also shows the assessments when they're due. So in week 6 we have a mid-semester quiz which will be held on the Thursday around the normal lecture time. Quizzes will cover the first 6 weeks of material, and we'll give you your grade back quite quickly on that quiz, and it'll give you a good idea of just what's required in the unit, what sort of level is required. And then later on, so one of the first practices we'll do is introduce this field project, which is a bird study, like I said. And throughout the semester, you'll conduct this study on your own in small groups, and then this needs to be submitted as a scientific article in week 11. And that's worth quite a large component of the unit. And yes, so you also notice that in week 13 we have an excursion to the Australian Museum planned. That will likely occur on the Tuesday, the normal prac day, but we could think about having that on another day. We'll work that out, the exact date of that suits everybody at a later time. And also during the last week, hopefully, of the unit, there'll be an opportunity to conduct some trapping and some spotlighting, and see if we can. If you want to come along, it'll be a voluntary thing. You can come along and see some of our local small mammals and bats and possums and birds in the field. We can get that organised. So what do I expect of you? As a general guide, you should spend around 10 hours a week in total over 14 weeks on this unit, okay? So that includes 5 hours of time in addition to attending all the lectures and prac classes. 5 hours per week in addition to the lectures and prac classes. That's an average level of effort as well. If you think you need extra study, or you wish to achieve a very high mark, which I hope you all do, that's what I expect, to be honest, then you should spend even more time on this unit. Okay, so 10 hours a week in total on the unit, on average, over the 14 weeks, or even more time. I really urge you, of course, like all lecturers I guess say this, but to attend all the lectures and prac classes. They're there for your benefit. So there's also a strong correlation I've noticed between attendance of the lectures and pracs and the students' mark in the unit. So if you can come along, please do take that opportunity. Remember to consult the View site regularly. I'll update that with announcements, and before you email me a question, please try to find that information first on the View site. With 140 students, I will try to answer your emails, but I really, if the answer is quite clear on the View site, I won't be that enthusiastic about answering your email, because you can find the information on the View site, and that's where I expect you to look first, okay? Remember, of course, that lab coats are always needed in the prac classes. There's nothing I can do about that. You need to bring a lab coat and wear it, or you can't be there, so please remember to bring your lab coat. Like I said, the book This unit is really based around this excellent textbook, Poe et al. Called Vertebrate Life. If you can afford it, buy one. There are also a whole bunch of copies in the library. Some of those are older editions, that's fine. An older edition is not a problem. If you can buy second-hand an older edition, that is also not a problem. I think there's some sort of scheme in the university where you can also get some help to buy textbooks. In any case, I do expect, to do well at this unit, you should be reading these chapters in that textbook. You should have access to that textbook. Whether you want to come to the library for a few hours every week and go through the textbook, or borrow the textbook, there should be some in the part of the library where they will always be there in reserve. In any case, that's the book. Assessments, like I said, you have the quiz, the mid-semester quiz. I just noticed before in the schedule that the weighting was incorrect, so this is the weighting for the assessments. I'll make that clear, but in the schedule I think it said 20%, but it's actually 25% for the mid-semester quiz and 30% for the scientific research article. I've submitted a request to change that, but it hasn't been accepted yet, so that's what was different in the schedule I showed you before. In the learning guide, this is what it says, and this is what it is. So the weighting is 25% for the mid-semester quiz, 30% for your research article, and of course then 45% for your final exam. So the mid-semester quiz gives you that opportunity to, get a good mark. Quarter of the unit's assessment is there, okay? So that's your real opportunity to get a good start for the unit. Yes, that'll be an hour long, the mid-semester quiz, held in week 6. Covers lectures and prac material as well, from weeks 1 to 5 inclusively. It's mostly multiple choice. That enables me to get feedback to you within a week. And it could have some short answer questions, but they certainly won't have long answer or essay questions. The second assessment is a research article. Like I said before, it's worth 30%. It's due in week 11, Friday the 29th of September at 5 p.m. And you submit that via views. Late submissions at this university always accrue a loss of 10% per day as per the university policy. Anyway, the task involves working in small groups of 3 people or less to conduct a scientific study to determine the factors that affect the diversity and abundance of bird species. As a group, you will develop your questions, you will design your study, and you will conduct your study and analyze the data, and then independently you will write up the study as a short scientific paper, i.e. of the format, a title page, abstract, keywords, introduction, methods, results, discussion, and references, as you would find in any standard scientific paper. So that's going to be fun, and that really enables you to come up with your own questions, to think about the design of the study, the sort of things you need to measure and conduct it. So I think that's a good assessment. Right, so let's get on to the first lecture. What's in this lecture? So first of all, we're going to talk about phylogenetics. And these are shared, we will discuss the topic of shared versus ancestral characters. Characters means a trait, a feature of an animal or an organism in general. The shared versus ancestral characters. And phylogenetics is all about hypothesized evolutionary relationships, which are what are described by cladograms. Then we'll move on to the phylum Chordata. So we will talk about which features, which characters are shared, derived characters that all chordates have in common. And then we will focus on the vertebrates, which are actually a subphyla of the phylum Chordata, the subphylum Vertebrata. That's where the vertebrates belong. So not all chordates are vertebrates, in other words. And then within the vertebrates, there are 2 classes based on whether the animal has a jaw or not, whether jaws have evolved or not within the vertebrates. And there are a few living, extant, that is, jawless vertebrates, the petromyzontiforms. And these are the hagfish and the lampreys. So they are examples of vertebrates that do not have a proper jaw. and they could be placed in the class Agnatha. And then we'll talk a little bit about then the rest of the vertebrates that have a jaw, that have evolved a jaw, and they are the Gnathostomata. And then the last part of the lecture we will focus on biodiversity of vertebrates, looking at some overall global patterns of biodiversity of vertebrate animals and some of the ecological functions of that vertebrate biodiversity. So phylogenetic systematics describes the evolutionary relationships among organisms. Central to phylogenetic systematics are what are called derived characters. These are features that are different from the ancestral form. A derived character is called an apomorphy. So apo means away from, and morph is form. So a derived character is called an apomorphy. And all species within a single evolutionary lineage share derived characters. That is, they all share characters that differ in their state from the common ancestor. And that's why they are defined, that's why they're in that evolutionary lineage. A shared ancestral character, that is, a character that is unchanged from the ancestral form, is called plesiomorphy, as opposed to an apomorphy. So plesiomorphy is a shared ancestral character which is unchanged from the ancestral form. Okay. An example of that is the vertebral column shared by all terrestrial vertebrates and lobe-finned fishes. This is a plesiomorphic character because these different groups all share it. It's this shared ancestral character. Right, so phylogenetics is based cladograms are a central feature of phylogenetics. So in determining the hypothesized evolutionary relationships based on shared derived traits, cladograms are constructed, and these propose the simplest phylogeny, the most parsimonious phylogeny, and that is one that assumes the least number of required changes in the process of evolution. Okay, so it maps out how different groups of vertebrates or organisms are related to each other via evolutionary changes, and it does that in a way that that assumes the least number of required changes in that process. The simplest phylogeny, or most parsimonious phylogeny. And here is an example. So take 3 taxa. Here they are here, 1, 2, and 3. And these taxa have 3 characters. They have scales, yes or no, so 2 and 3 have scales. They have toes of different numbers, 4 or 5. So 1 and 2 have 5 toes, and 3 has 4 toes. And they have tails. They could either have a tail, such as number 1, or not have a tail, such as 2 and 3. And the question is, what evolutionary steps might have occurred to lead to these different organisms? How can we make a cladogram that describes the simplest evolutionary pathways that would have led to these organisms? Only 3 changes are needed to produce the derived character states. The first change is that scales are lost. The second change is that the tail is lost. So these 2 organisms now lack a tail. That's the second change that has occurred. And the third change that is required is that organism number 3, there's a toe has been lost. So it goes from 5 toes to 4 toes. So this is the cladogram showing the evolutionary relationships between these 3 organisms that has the least number of assumed evolutionary events. 3, in fact. So this is the simplest, or most parsimonious phylogeny. 2 other phylogenies, i.e. cladograms, are also possible, B and C. But each of these requires a tail loss to have occurred twice, here and here in this case, here and here. And an evolutionary scenario assuming a larger number of steps, 4 steps in these 2 cases compared to 3 here, is less likely than a simple evolutionary pathway with fewer steps. So although these cladograms are possible, these hypothesized evolutionary relationships are possible, they're less likely than this one. And so this is the assumed evolutionary relationships, or cladogram, because it's the simplest one. However, a phylogeny, like other scientific hypotheses, is always open to be changed. It's not fixed, it's just an hypothesis. And given data that does not support the most simple hypothesis or any other cladogram, the proposed relationships can be changed. It's based on the new evidence, okay? It's open to change. So as I just said, cladograms depict hypothesized evolutionary relationships among lineages or clades of organisms. And the structure of cladograms is based on shared derived characters. So organisms within a lineage or clade share a character that has derived from the ancestral condition. It's differed from the ancestral condition. And branches in the cladogram, so here, these branches, they depict points at which clades have diverged because of the evolution of a change in some feature or character that is shared by all members within one clade, but not the other one, but not the lineage outside the clade. For example, at number 1, with a vertebrata, all vertebrates possess vertebrae, whereas the lineage outside this branch do not possess vertebrae. Number 2 describes the branch point whether all organisms after that branch point can be referred to as anathostomata. They all have jaws that are formed from a mandibular arch, whereas organisms in their lineage, the other lineage that's not part of anathostomata, are the jawless vertebrates. So they're within the vertebrates, but these are the jawless vertebrates, and these are the vertebrates that have jaws. 3, the osteichthyes. These vertebrates have, or these jawed vertebrates have a shared derived character, and that is the development of a lung or a swim bladder, which is derived from the gut, and also a unique pattern of dermal bones in the head and the shoulders. These are, those features are shared by all organisms, all vertebrates within the osteichthyes, but not the chondrichthyes, sharks and skates and rays. Next along the line, after osteichthyes, are the sarcopterygiae. And so that point in the evolution of vertebrates is where vertebrates have developed a supporting skeleton in the fins. The next step, number 5, the rhipodistia, are where teeth have evolved with enamel and certain unique patterns of dermal bones in the skull. So all organisms within that lineage share those derived traits. And then after that are the tetrapods. These organisms within this lineage have limbs with carpals, tarsals, and digits. And after that, there's the next branching point, is the distinction between the amniotes and the non-amniotes. So the amniotes are those organisms that share the derived trait of embryo-derived membranes in the egg, the amnion, the chorion, and the allantois, which are embryo-derived membranes in the eggs. And these are largely terrestrial animals. Okay. Note that cladistic groupings are nested progressively, so that all eutherian mammals are therians. So all of these eutherian mammals are nested within Eutheria. and all Theria are nested within Synapsida, and all synapsids are amniotes, and all amniotes are tetrapods, etc. Okay? So cladistic groupings are nested progressively. So now if we just focus in on the archosaurs, this group here, look a bit more detail at the cladogram for this group that's shown here. Archosaurs are here, and here's their sister group, Lepidosaurs. The assumption that organisms within a lineage of a cladogram share characters that are inherited from a common ancestor allows inference about the characters of extinct species within those same lineages. For example, birds and crocodiles share the character of strong parental care of their young, let them look after their young. Whereas this behavioural character, is rare in lepidosaurs, lizards and snakes. It's not so common to look after their young. And lepidosaurs have a sister lineage to the archosaurs, okay? And the most parsimonious explanation for this situation assumes that parental care is an ancestral character of the archosaur lineage. That is, it has evolved before the split between crocodiles and dinosaurs and birds. So it's a character trait that's probably shared by all archosaurs. And therefore it can be inferred that other groups of archosaurs that are now extinct, such as the dinosaurs, were also likely to have exhibited parental care. The parental care we know is in birds and crocodiles, which are both living or extant species or groups. And there's no parental care in lepidosaurs, it's rare. So you can infer the parental care was shared by the common ancestor of all archosaurs. And then that suggests the most simplest hypothesis is therefore that extinct archosaurs, such as the dinosaurs, also probably had parental care. So that's lineages that share derived characters can be used to infer about the state of unknown characters in extinct species as well. Okay? Right, a little bit more about some terms to do with phylogenetic relationships. Some characters can be called homologous characters, and these are characters that were derived from a shared ancestor. So, for instance, the bones in the forelimbs of tetrapods are all homologous. So the same bones within the forelimbs of different tetrapods all came from a shared ancestor. They're homologous traits or characters. Analogous characters are different. Analogous characters look similar, so they're similar in form or function, but in fact they were not shared. They were not present in the last common ancestor of those groups. And this resemblance of characters that are actually not homologous, that are analogous, this occurs via a process of convergent evolution, where the same sort of solution has evolved in the face of selective pressure, in some environmental selective pressure, but that character has evolved from different traits, not from a shared trait. So although the bones in the forelimbs of tetrapods are homologous, the wings as a structure are not. So in fact the wings have evolved separately in pterosaurs, type of dinosaur, bats and birds. So the common ancestor of these three groups did not have wings, but the three groups evolved wings separately to solve the problem of flying. And so the wings of these three groups are analogous characters, not homologous characters, and they look similar in form and function because of a process of convergent evolution. So homologous versus analogous characters. Right, okay, so now it's time to look get a bit of a historical perspective on history of Earth and where vertebrates fit in terms of fossil evidence for their evolution. We know that the Earth is around 4.5 billion years old. And the first organisms found as fossils occur around 3.5 billion years ago. And these are microbial mats of bacteria and other sorts of protozoans, including photosynthesizing organisms, apparently. But the oxygen really there's evidence of the oxygen containing considerable amounts of the atmosphere containing considerable amounts of oxygen from around 2.4 billion years ago. So clearly photosynthesis was occurring before that, ramping up, and now oxygen is detectable in the atmosphere from around 2.4 billion years ago. These are all single-celled organisms, however, before that, and the eukaryotes, which are organisms with more complex cells, with mitochondria and organelles, real step up from prokaryotics. The eukaryotes evolved apparently around 1.85 billion years ago. So around here, eukaryotes. And then multicellular eukaryotes from around 1.7 billion years as well. Terrestrial plants arrived on the scene 450 million years ago. There was some evidence, or fossil evidence, of land plants in this part of the Paleozoic era, 450 million years ago. But vertebrates arrived on the scene a little bit earlier than that. The first vertebrates were apparently evolved around 525 million years ago, in the Cambrian period of the Paleozoic era. Tetrapods on land evolved. First fossils of those found a little bit earlier than 360 million years ago. Up here, start of the Carboniferous. You see a radiation of amphibians, sharks. So the first tetrapods, the amphibians are formed there. And then the amniotes, which is an essential adaptation. Those vertebrates have these embryo-derived membranes in the shells. There's evidence of them evolving around 330 million years ago. All right. And then if you just look at it more closely, in the last 66 million years, approximately when Australia broke free of Antarctica, for example, and started to drift northwards, following the large extinction event between the Cretaceous and the Tertiary period. That was when there was a diversification of the mammals and a lot of good fossils for birds and mammals from then on. Right. So there are 5 derived characters that are shared by all chordates that really define the phylum Chordata. Those 5 derived characters are one, a notochord, which is a dorsal stiffening rod here, which in most vertebrates is replaced by the vertebral column. 2, a dorsal hollow nerve cord on top of the notochord. 3, a gland called an endostyle in early vertebrates, which is homologous with a thyroid gland in more derived vertebrates. And the endostyle is a ciliated groove used for feeding in the cephalochordates, and it's, like I said, homologous to the thyroid gland of vertebrates, which regulates metabolism. 4, a muscular post-anal tail, so a tail that contains segmented muscles that extends beyond the gut. And 5, a pharynx with slits in it, gill slits, here in the pharynx region. So the openings in the pharynx are used for filter feeding in the cephalochordates, but gas exchange in all other vertebrates. So, they're the 5 derived characters that define the phylum Chordata. But where do the chordates, the phylum Chordata, where does it fit among other mammals? So the chordates are one of around 30 animal phyla. Outside of the chordates, we vertebrates are most closely related to echinoderms, which include starfish, sea urchins. So you're more related to starfish and sea urchins than you are to any insect, worm, or mollusk. It seems a bit strange because chordates superficially resemble other bilaterally symmetrical active animals like insects, but there are strong and clear developmental and molecular evidence that point to a relationship between the chordates and the echinodermata as the nearest relatives of the chordates. The chordates, the echinoderms, and two other phyla are what are known as deuteriostomes. All other phyla of animals are protostomes. So both deuterostomes and protostomes are coelomet animals. They have 3 tissue layers in their body, and they have a body cavity within the mesoderm. But deuterostomes are different because what that means is that at the gastrular stage of embryonic development, and there's a cellular ball and a little indentation in that cellular ball, that first opening, in this, it's called the blastopore, that becomes the anus, and the second opening becomes the mouth. In contrast to the protostomata, that first opening in the protostomata becomes the mouth. Okay? So echinoderms and chordates and 2 other phyla are deuterostomes. In the deuterostomes, the first opening, the blastopore, becomes the anus, and the second opening becomes the mouth. The Vertebrata is, like I said, a subphylum of the vertebrate Chordata, and 2 other subphylum are also included as chordates, the cephalochordates and the urochordates. It's shown here. So here's the chordates, all these grouped here. But these ones are the vertebrates. There's 2 other chordates that are not vertebrates, cephalochordates and the urochordates. And these are colloquially known as the protochordates. Let's have a little bit of a look at those first. So the urochordates, subphylum Urochordata, these are known as tunicates or sea squirts. And the adults of most living extant species are sessile, they're non-moving, and they're filter feeders. They look like this. Water comes in here, it's sieved by this pharynx. They sort of filter feed through their pharynx, and the water then goes out there. The food is collected in sort of mucus and goes down to the stomach and out the anus there. However, this doesn't look much like a vertebrate, right? Or like a chordate, sorry, because they're not vertebrates. They're chordates. So where are these features that we talked about that all chordates share? Well, they're found in the free-swimming larval stage of tunicates, of urochordates. So here's a larva, free-swimming larva, and here's the notochord, post-anal tail, the dorsal hollow nerve cord, the pharynx slits in it, and endostyle is there as well. So most tunicates have these free-swimming larvae which last hours or days, and the larvae share or exhibit the shared derived chordate characters that we talked about before. And the ancestral state of all chordates probably resembled this free-swimming larval tunicate. The other protochordate is the cephalochordates. These are known as Amphioxus or lancelets. They possess these myomeres or blocks of striated muscle fibers along their body. They're also filter feeders. They lodge themselves in the substrate like this and pump water through their pharynx and sieve off bits of food. That food, so water comes in here. It gets sieved through their pharynx, holes in their pharynx, endostyle, produces mucus, which captures that food and delivers it down into the gut. They have the notochord is here, the stiffening rod. The encephalochordates actually extends right up through to the head, gives a stiffening ridge right up through the head, and allows them to sort of wriggle their body from side to side, might help with burrowing. They don't have any gills. The slits in the pharynx serve for feeding, not for gas exchange. They just simply absorb CO2 and exchange gas through their body because they're so small and not very active. And they're a filter feeder, like I said. And they have the 5 features that are shared by all chordates. This brings us to the subphylum Vertebrata, and the defining feature of vertebrates that separates them from the other chordates is that they have a cranium, some sort of bony, cartilaginous or fibrous structure which surrounds the brain, some sort of protective structure around the brain, a cranium. That's the defining feature of all vertebrates. In most vertebrates as well, a bony vertebral column replaces the original notochord after the embryonic period. Although in some fishes, the vertebrae are made of cartilage, of course, rather than bone. Vertebrata, what are the features of vertebrates? They have some unique embryonic features. The Hox gene, which controls the development of a body plan. Vertebrates differ from protochordates and other invertebrates because of a number of features, including the fact that they have multiple copies of this Hox gene. Have a neural crest, which is a new embryonic tissue, i.e. a germ layer like the ectoderm or the endoderm and mesoderm, and it forms novel vertebrate structures. Placodes is another type of embryonic tissue that is unique to the vertebrates, and placodes are epidermal thickenings that give rise to complex sensory organs. MicroRNAs are another unique embryonic feature of vertebrates. MicroRNAs are non-coding RNA sequences, and vertebrates have microRNA that regulate the development of unique structures in their body plan. The earliest vertebrates had derived characters that give them important advantages over their non-vertebrate chordate ancestors. Most conspicuously, vertebrates have a distinctive head end containing a tripartite brain, now covered with a protective cranium and linked to a set of or a bunch of sensory structures, nose, ears, eye, and inner ear. They use a muscularized pharynx and gill skeleton to pump water across their gills. That's different from the non-vertebrate chordates. And they use their muscular pharynx to pump water over their gills for gas exchange rather than for filter feeding. And they've now developed a better pump, a heart, a 3-chambered heart, and a closed circulatory system to move these gases around their more metabolically active tissues in their bodies, which include powerful muscles in the tail. So the early vertebrates were likely to be active predators rather than passive filter feeders. So this table summarizes those differences between the vertebrate chordates and the non-vertebrate chordates. And so the vertebrates in the notochord stops before the head, ends at the head, sorry. They have a cranium surrounding the brain, which is tripartite. The brain is there's 3 parts to it, rather than a simple brain than in the non-vertebrate chordates. Their sensory organs are more advanced. Their gill arches are used for gas exchange, which is for respiration rather than for feeding. The pharynx has become muscularized, enables them to really suck in water into their pharynx much more strongly, as opposed to the ciliary action in the pharynx of non-vertebrates. Water movement is via the muscularized pharynx rather than ciliary action, like I said. The gut is also muscularized. Digestion occurs extracellularly. So there's a stomach where digestion can occur outside of the cells, which allows much more effective digestion. There's a three-chambered heart, a closed circulatory system, including a dorsal aorta to pump around that gas to where it's needed. And the blood now contains special pigment that allows to carry the oxygen throughout the body, which does not occur in the non-vertebrate chordates. One feature that the earliest vertebrates did not have is jaws. These vertebrates, the jawless vertebrates, are called agnatha. The first vertebrates were jawless, agnathians. Agnathans include several extinct groups, as well as two groups of living species. The hagfish and the lampreys are the living jawless vertebrates. They're also colloquially known as cyclostomes or round mouths. There are several extinct groups that are very strongly apparent in the fossil record, including the ostracoderms, which were probably more closer to the jawed vertebrates than the living jawless vertebrates. So even though hagfish and lampreys species still occur, they probably represent quite ancestral form of the vertebrates. So like I said, the vertebrates can be divided into these two groups, the jawed or gnathostomes and the jawless agnathans. And agnathans include the living varieties, the hagfish and lampreys, which are colloquially known as the cyclostomes, and some extinct forms, most important of which are the ostracoderms. So let's cover the extinct form first. The ostracoderms, even though they're more derived probably than the lampreys and the hagfishes. These guys were most common in the Paleozoic 500 million years ago. They're jawless fishes. They're pretty much encased in body armor. That's why they're fossilized so well, in contrast to the lampreys and hagfishes, which lack bone altogether. Ostracoderms are characterized by the presence of this mineralized bony tissue located within the dermis. So just to have it underneath the epidermal skin, within the dermis, is this mineralized bony tissue. It's especially prominent in the head region, where it forms a shield, where it can form an armored shell or a shield called a carapace. The function of this bone is not 100% clear, or the hypothesized function. It could be that it served as some form of protection. Some of these spines seem to suggest that was necessary, or certainly would seem to serve that kind of role. But there are other potential functions. It could actually be a place to store calcium and other minerals. It also possibly could serve as insulation around electroreceptive sensory organs, or possibly allow these early fish to sort of fly better through the water, have a sort of hydrodynamic function, to give them lift while moving forward in the water. So like I said, ostracoderms are actually more closely related to the gnathostomes, the jawed vertebrates, than they are to the living agnathans, the hagfish and lampreys. Getting onto those living jawless vertebrates, the hagfish, the myxinoforms, they're basically eel-shaped animals, free-swimming. Most of them are scavengers of large animal carcasses, such as whales and things that sink to the bottom of the ocean. Very small paired eyes, sort of tentacle barbels around the mouth, and horny plates in the mouth and on their tongue, which contain teeth made of keratin that are used for scraping flesh. The scavengers, so they also eat some worms. Very simple digestive system, which is typical of the scavenging, and they can also absorb some nutrients over the skin, and they have this characteristic of defensive mechanism of producing copious amounts of slime all of a sudden, and that can readily fill the mouth of a small shark that might want to eat them, and it's an effective anti-predator mechanism. Lampreys are also eel-shaped. Lampreys are petromyzontiforms. They're also mostly parasitic now rather than scavengers. They attach to the host with this sort of suction-like mouth, and then they scrape away the flesh, and then they feed off the blood of the host. As a parasite, their gastrointestinal tract lacks a stomach. It's very simple. It just needs to absorb those nutrients, basically. On the side of the body, they have these gill pouches. They're blind ending. They don't go into the pharynx. So they use a type of in-out tidal ventilation. It's probably an adaptation that allows them to remain with their mouth completely stuck onto their host and still exchange gas or exchange water, allow gas exchange with these in-out flow of water through their gill pouches, and they have a single nostril on top of their head, nasohypophyseal opening, which is the sensory organ on their head. Yeah. I have a little video about lampreys, which I'd like to play for you if it works.
To find your ideal hotel, just go to Trivago, type in where you want to go, select your check-in and check-out dates, and search. Remember, Trivago...
Sea lampreys usually attach to larger fish in the ocean to feed, but recently it seems they've begun to attack swimmers too, who've dared to venture into the lake's deeper waters. Dr. Ellen Marsden has become a specialist in these resilient bloodsuckers, which are commonly confused with eels. But this is by far the most ancient river monster I've ever attempted to catch. Seeing eel and lamprey side by side, the difference starts to become clear.
This is a true vertebrate. This is a proto-vertebrate. It's just in the process of becoming a vertebrate. It doesn't have a bony backbone. It has no bones in the body at all. It is so primitive it doesn't even have a true jaw. It has no lower mandible. It can't bite, whereas an eel has a true jaw and can actually bite into something. So it's a fish, but it's the very beginning of the fish lineage.
Around 300 million years of evolution separate these two animals. In fact, we have more in common with the eel than the eel does with the lamprey. So the predation is all about this structure on the front, is it?
Right. And it's rather deceptive because you think, wow, all those teeth. They are teeth, but not in the way we think of them. They're not to bite. These are just grippers. It's like having gripper gloves to be able to grab something that's slimy. The thing that does the damage is actually down here in the mouth itself. So it has a tongue, much like the eel has a tongue, and on that tongue are chisel-like teeth. So this tongue is acting like a piston. It's going to come out and actually it rasps a hole in a fish. And again, it can't bite, so having rasped the hole into the fish, it's then going to use suction to suck body fluids, blood and other material out of that hole that it's just made.
The next image that I saw.
Alright, so there you go. So the major difference between the agnathans and the gnathostomes are the presence of a jaw in gnathostomes with teeth in two sets, and also in addition gnathostomes have two sets of paired appendages, the pelvic and pectoral appendages in the gnathostomes. However, the gnathostomes, the jawed vertebrate body plan differs from that of the jawless vertebrates, the agnathans, in many other important ways. And basically, overall, the gnathostomes are much more active and complex vertebrates. So this is a generalized body plan showing the derived features of gnathostomes, jawed vertebrates that differ from the jawless vertebrates. So first of all, like I said, the gnathostomes now have these jaws that are formed from the mandibular arch with embedded teeth. And they have two pairs of paired appendages, pectoral and pelvic, and fish have fins. They have a gill skeleton, which consist of jointed branchial arches and enhanced gill muscles here in this pharynx region. They also have hypobranchial musculature, so muscles in the branchial region, and combined with these branchial skeletal elements allow for suction, feeding and breathing by opening their mouths and their pharynx very suddenly with these muscles and these bones here. These bony elements or cartilaginous elements enables a strong suction both for sucking water, for pushing across the gills, and also for feeding. We'll get to that later. They have two distinct olfactory tracts with two nostrils. The original first gill slit that after this first set of gill arches becomes the jaw, this original gill slit in some fishes becomes a spiracle. And the spiracle actually allows the intake of water into the pharynx when the mouth is otherwise occupied, such as in skates and rays. We'll see that later. They have three semicircular arches in their inner ear. So in addition to two, they now have a horizontal canal as well. So the inner ear is all about balancing, right? Now they have three semicircular arches in the inner ear. They have in addition to the heart, the conus arteriosus, and this occurs between the ventricle and the ventral aorta, this region here. And what it does is it smooths the pressure caused by a more powerful beating of the heart, this flexible section. 8 have a lateral line or horizontal septum dividing the muscles into the dorsal and ventral sections. And along this is the position of the lateral line, which is a sensory system containing neuromast cells. And they have vertebrae, and the vertebrae have a centrum which surrounds the notochord and ribs. And together these features result in improved locomotion and the sensory organs on the head, the jaw and the tail, allow these animals to be much more active and to become predators, in fact. So the jaw, this is a major evolved trait, key feature of the gnathostomes. It's hypothesized to have evolved from the gill arches. So it's thought that the first gill arch formed the mandibles. Here is this ancestral condition of the jawless vertebrates with the gill arches and the slits in between them. And here's the brain case, the cranium above it. And it's thought that this first gill arch here becomes the mandible, and the second gill arch forms this hyoid arch which supports the jaw. And this first gill slit, like I said, becomes a spiracle. So their jaws, their mandibular and hyoid arches evolved from the first and second gill arch of the jawless ancestral vertebrates. Also can be seen here in the actual skeleton of the lamprey, which is a living jawless vertebrate. And so here you can see the gill arches and the pouches. And then this is the pharynx region of a shark, and you can see the mandibular arch and the hyoid arch here. So these are thought to have derived from the first and second gill arches of the jawless vertebrates. But there's a question, however, in that what could have been the function of the jaw, the evolving jaw, before it was really effective as a jaw? So what use is a proto-jaw that doesn't really allow biting, but is evolved halfway between that of a jawless vertebrate and a vertebrate? And it seems one of the most likely hypothesized function that could have allowed the jaws to develop into sort of into a biting mechanism was that if the jaws were initially used not so much for biting but for increasing the strength of gill ventilation. So gnathostomes are very active and are more active than the jawless vertebrates, and they have high metabolic rates, so they do require more efficient gas exchange with the environment. So the mandibular arch, this first has evolved from the first gill arch, could have become enlarged, or did become enlarged into this proto-jaw, and that may have been because it allowed more forceful gill ventilation by allowing sort of snapping, rapidly opening, and the increased musculature of the pharynx as well would allow that to sort of snap open and draw water into the mouth much more strongly. And then it also would allow the mouth to close much more effectively and push that water across the gills. And that type of musculature suction breathing, supported by these stronger and different shaped arches, may have also facilitated suction feeding. So suction feeding is very common among fish. It's where predatory fish creeps up to the prey and then rapidly increases the space within its pharynx by snapping open its mouth region, its pharynx, and draws prey into its mouth. So this suction respiration could have also allowed suction feeding and led to the more derived function of the jaw as a way of grasping things, of biting. So the function of a proto-jaw could have been for increasing the strength of gill ventilation first. So now we have the jawed vertebrates. They can grasp prey and have this powerful suction from the branchial muscles and high levels of activity. And the jawed vertebrates can be placed into three major groups: the placoderms, which are all now extinct, and two extant groups: the cartilaginous fishes, chondrichthyans, and the bony fishes, osteichthyes, and one group of bony fishes, the lobe-fin fishes, sarcopterygians, are the ancestors of all the tetrapods. Let's have a look at the placoderms, which are the extinct group of jawed vertebrates. So they had jaws, but they're basal to other forms of jawed fishes. Paired pelvic and pectoral fins, a sort of armoured head, which was separate to the back of the body, and they seem to be mostly predatory species. Those sort of bony plates instead of real teeth. Yeah. So they are the placoderms. So this shows the classification now of all living fishes. So we're in the phylum Chordata. We're looking at vertebrates. So there are subphylum within the Chordata that have a cranium, most of which also have vertebrae. And like I said before, can be into two groups: the agnathans, the jawless vertebrates, of which living now are the lampreys and hagfish, and the jawed vertebrates, the gnathostomes. And the gnathostomes can be broken down into two groups: the cartilaginous fishes, the chondrichthyes, and the bony fishes, the osteichthyes. So within the chondrichthyes, we have the sharks and rays, the elasmobranchs, and the holocephali, which are the ratfish. Those two groups are in the chondrichthyes. And in the bony fishes, the osteichthyes, they can be separated into two groups: the Actinopterygii, the ray-finned fishes, and the Sarcopterygii, the lobe-finned fishes. And we can really, or all tetrapods can also be grouped within this Sarcopterygians, ancestors of all tetrapods. Right. So that is the point that we're going to leave it for now in terms of the evolved features of the vertebrates. Now we're going to move on and look at some aspects of vertebrate biodiversity, the second half of this week 1 lecture. Each of the 7 vertebrate classes, the Agnatha, the jawless fishes, the Chondrichthyes, the cartilaginous fishes, the Osteichthyes, the bony fishes, the amphibia, the reptiles, the aves or birds, and the mammalia, the mammals, will be covered in lectures in the coming weeks. Of the 63,000 living vertebrate species, around half of all those species are fishes, and most of those are bony fishes, belonging to a single lineage, the Teleostei, of which there are around more than 30,000 species. However, the smaller groups of cartilaginous fishes are very important because they often, like sharks, often play an important role in ecosystems as apex predators. Even though there's not very many species of them in terms of diversity, they do often play important roles in ecosystems, the cartilaginous fishes. The second largest class are the birds, with around 10,000 species, with around 8,500 species of reptiles, which are made up of crocodilians, snakes, lizards and turtles, around 6,000 species of anura or amphibians, and a little bit less of mammals. So what is biodiversity? Can we define it? What's the definition? One definition is that biodiversity is the variation among genes, species, and functional traits. It's often measured as richness, which is a measure of the number of unique life forms, or evenness, and that's a measure of the equitability among life forms. So is this richness made up of mostly just one species and a whole bunch of things at a very low level, a whole bunch of other life forms at a very low abundance? Or is it made up of roughly equal numbers of different life forms? That's evenness. And it can also be measured in terms of heterogeneity, and that describes the dissimilarity among life forms, i.e. that biodiversity might be made up of vertebrates, mammals, fishes, reptiles, other animals, or it may be made up of just one group. Okay, so you can talk about richness, so number of different species or life forms, evenness, the relative abundance of those different life forms, and heterogeneity, how similar those life forms are, typically from an evolutionary phylogenetic perspective. Animals are not obviously evenly distributed around the Earth. In fact, they can be early on. It was worked out that they can be partitioned into different regions, different groups around the Earth. These biogeographic units are really fundamental to understanding evolutionary processes on the global scale. And they're also fundamental for driving conservation of biodiversity. The person that really came up with, who started this area of biogeography is a guy called Alfred Russel Wallace. Wallace, of course, you should have heard of already. He's a co-founder of the theory of evolution by natural selection with Charles Darwin. They co-wrote the paper, the first paper that proposed the theory of evolution by natural selection. And Alfred Russel Wallace published the first map of global terrestrial zoogeographic regions. So he's therefore known as the father of biogeography. And there was a paper out recently, this one, an update of Wallace's zoo geographic regions of the world, and they reanalyzed, integrated all the data they could find on current global distributions of species and also the phylogenetic relationships among those species. And they used the distributions and the phylogenies to derive 11 zoo geographic realms, and they are shown on this map here. They also compared, like I said, the phylogenetic uniqueness of these different realms and regions. And that's shown in this figure. This shows the pairwise phylogenetic beta diversity. So the diversity among habitats, taking account of the evolutionary relationships between organisms. And they use that to quantify the change in phylogenetic composition among species assemblages across the globe, and it could also be termed evolutionary uniqueness. And then, so this phylogenetic uniqueness of regions is with the Australian, Madagascan, and South American regions being the most phylogenetically distinct, or having the most phylogenetically distinct assemblages of vertebrates. The darker this colour, the higher this number. Those regions, those realms, contain the most phylogenetically different assemblages of vertebrates. These evolutionary unique regions harbour radiations of species from clades that are either restricted to a given region or found in only a few regions. Okay, so the diversity of vertebrates can be broken up into these 11 zoo geographic realms, but they're not all equal. And you can look at it from a phylogenetic perspective to look at the uniqueness of these different groups. There's Alfred Wallace. He was famous for discovering, also he was most famous really for discovering a remarkably distinct break in the composition of vertebrate species among the islands of Southeast Asia. This break in the diversity, or the types of different vertebrates you get between Southeast Asia and Australasia, is known as Wallace's line. The exact position of the line has been modified here and there, but in any case, it stretches in this sort of deep oceanic, this deep channel between Bali and Lombok, between Borneo and Sulawesi. On the Asian side of Wallace's line, you get things like primates and cats, and ruminants, and flying rodents, and hornbills. In terms of birds and mammals, deer, rhinoceros, tapir. And on the Australian side of this Wallace's line, you get completely different suite or distinctly different groups of animals. So you get monotremes, like echidnas, you get the marsupial macropods, the marsupial possums, and you get the ratites, like the cassowary. You get cockatoos, you get strange noisy birds like friarbirds, honeyeaters, you get gliding marsupials. The explanation for this abrupt boundary between lineages of vertebrates is, of course, continental drift, a theory which was first proposed in 1912 by Alfred Wegener, which was 36 years after Wallace's publication on this line. But in fact, continental drift was only accepted relatively recently. It only was accepted as irrefutable theory in the 1960s. So Wallace didn't know why there was such a strong break here, but the underlying reason is that vertebrates and other animals and plants evolved in Australia and Australasia, in this part of the continent, as it drifted slowly up from Antarctica. Evolved separately as a big island before it bumped into Southeast Asia. It's Wallace's line. Yes, so like I said, the explanation for the evolution of geographic patterns in distinct groups of organisms is continental drift. So during the evolution of the vertebrates, the continents have moved around the place, moved around the globe, joining and breaking up and experiencing different latitudes, ocean currents and climates. Around 300 million years ago, all the continents were joined in a supercontinent called Pangaea, which was the birthplace of terrestrial vertebrates. This supercontinent began to break up around 150 million years ago, here and here, and it into two major continents, one called Laurasia and one called Gondwana in the south, and then it broke up into further smaller units. Isolation of different lineages of vertebrates occurred because they evolved on these land masses that were isolated from each other, although they sometimes came together again when these land masses collided. And the moving around of the continents via continental drift can explain a lot of the patterns in, well, not all of the patterns, but underlies the global patterns in these zoogeographic realms around the globe. One of the oldest known and most fundamental patterns in biodiversity, in global biodiversity, is the increase in species richness that occurs at lower latitudes, closer to the equator. So here shows the distribution of mammals around the globe. It's shown here as well. So you can see on this figure, the species richness is shown on the y-axis as a function of latitude. You get many more species at the lower latitudes near the equator than you do as you go further away from the latitudes. And you find the same latitudinal gradient in species diversity also in birds, amphibians, and virtually all other taxa, or many other organisms, both invertebrate and vertebrate. There are a large number of hypotheses that have been proposed to account for this very strong pattern in global biodiversity of this latitudinal gradient. And these are some of the names of these hypotheses. Abiotic, biotic hypothesis. Well, these hypotheses can be kind of grouped according to the likely explanatory mechanism, and that's how they're shown here. So whether they're related to the amount of energy available around the globe, evolutionary rates, evolutionary speed, differences in geographic area, population dynamics, productivity, time. There are lots of different hypotheses proposed to account for latitudinal gradients in biodiversity. One that I want to focus on, which I think is strongly supported by the evidence, although is definitely not accepted by everybody at all at this stage. But in any case, I think it's an important hypothesis, and it's also was proposed by a professor at a university where I did my degree, University of New England, Professor Klaus Roeder. And this hypothesis is called the hypothesis of effective evolutionary time. And it suggests that species have accumulated fastest in areas where temperatures are highest. It's known that mutation rates and speed of selection occur faster due to faster physiological rates at high temperatures. And generation times, which also determine speed of selection, are smallest at higher temperatures. So this leads to a faster accumulation of species at higher temperatures, which are absorbed into the abundantly available vacant niches in the tropics. So niches are available, Professor Roeder suggests, at all latitudes, and differences in the number of such niches cannot be a limiting factor controlling the number of species you fit into each ecosystem. So Klaus Roeder really thinks that there are available niches, that niche availability is not a limiting factor, and it's just related to the speed at which species have evolved in the tropics. Combined with the faster rate of species evolution in the vacant niches is a time factor. habitats with a long, undisturbed evolutionary history, which is the case for many tropical environments, will have greater diversity than habitats that are exposed to disturbances in evolutionary time. So, low latitude environments were largely immune from, or were more protected from things like ice ages, and have had a very stable history. So that and the faster mutation rates, speed of selection, and smaller and shorter generation times, Professor Rhoda suggests, has led to the latitudinal gradient in species diversity. It's still an open question, a very interesting thing. Right, so there are two global patterns in global species diversity that we've just covered. Why is biodiversity important? Well, it has intrinsic value, so it's the result of millions of years of evolution, so we can value it just for that alone. It also has aesthetic value. Its natural heritage may have some sort of spiritual or cultural value and recreational value. It could have iconic value, it could have some special value, i.e. the koala, for example. Or it could be useful to us, it could be a source of food or clothing, or it could provide a medicine. It could be a pest to humans, such as starlings. It could be important because it's a pest. It could also be important because it serves key functions in an ecosystem to maintain the function of the entire ecosystem. And those functions could be to allow primary productivity, could promote the cycling of nutrients within that ecosystem, the formation of soil, pollination of crops, the purification of water, the regulation of climate and disease. One thing to keep in mind, though, is that most animal biodiversity is represented by the invertebrates, okay? So the size of these different organisms here is representative of how diverse they are. Most things are actually beetles, most animals are in fact beetles, and vertebrates are not very diverse in the greater scheme of things. But vertebrate biodiversity can be important because of unique features of vertebrates. They are large, they are very active, metabolically active, and they can have some major effects on ecosystems. And two examples of the effects of vertebrates on ecosystem function that we'll focus on are something called trophic cascades, and they are where apex consumers, apex predators can have effects on other animals at different levels, different trophic levels, indirect effects as well as direct effects, and ecosystem engineers. So, trophic cascades. Trophic cascades are where the presence or the absence of an apex consumer has direct effects on lower trophic levels, i.e. by consuming prey, but also wide-ranging indirect effects because of species interactions. For example, sea otters enhance kelp abundance by limiting herbivorous sea urchins. So therefore they have an indirect effect on interacting species. So the sea otter eats the urchins, and the sea urchins are herbivorous and they eat the kelp. So the presence of the sea otter can enhance the kelp in the ecosystem by eating the sea urchin. So there's an indirect effect of the otter and the kelp. And that's why it's a trophic cascade. And the loss of these apex consumers has pervasive effects on natural ecosystems. So it's been found that where sea otters are more abundant, there's less sea urchins and more kelp, like I said, so sea otters have a positive benefit on kelp. But this has even further, more wide-reaching effects. So where sea otters are present, there is greater level of fish abundance. And because of the increased amount of fish, there's more fish in the diet of gulls. For example, there is more muscle growth where sea otters are present, and the diet also affects the diet of things like eagles. So, it has these flow-on effects that go through and across different species and that are caused by the interaction among species. So a single, in this case, vertebrate apex predator can have multiple effects on different parts of the ecosystem. Here's 4 more examples that show these trophic cascades. So this has got to do with a consumer, an Arctic fox. Where the Arctic fox is present, the grasslands turn back to tundra because the foxes limit the number of seabirds and reduce the soil nitrogen inputs. So when you have the Arctic fox, that environment looks like this, and when they're absent, you have more seabirds, a higher level of nutrient, and the ecosystem changes to more of a grassland. Where jaguars are absent, they have reduced plant survival because there are more herbivores. So the jaguars reduce the numbers of herbivores and prevent the sort of damage to the environment that herbivores or the herbivory that those herbivores cause. Similarly with the wolves, where the wolves are present, there is enhanced willow growth because of reduced grazing by the elk, by the deer. You have more wolves, less deer, more vegetation. Wildebeest, this has got to do with a disease. You have a decline in the woody vegetation when you have more wildebeest. In the Australian system, dingoes are an apex predator where they are present, and the presence of dingoes seems to have wide-ranging indirect effects on the ecosystem as well. So, for example, it's hypothesized that dingoes can have a suppressing effect on foxes and cats. So where you have increased numbers of dingoes, you may have greater numbers of the prey of foxes and cats, i.e. small goannas and mice and small mammals. So higher numbers of dingoes, higher numbers of the prey of their prey. And similarly, dingoes can suppress herbivores such as macropods, and if you have fewer numbers of macropods, you can have a change in the vegetation, increased density or complexity of the vegetation. So you take out a single vertebrate apex predator, and that will reduce their numbers, and that can have flow-on effects for other parts of the ecosystem. Another example here is shown with rabbit control. I might talk about that during the lecture itself. It's a bit easy to explain, so skip that for now. Ecosystem engineers is another type of function that vertebrate animals can have in an ecosystem. And that is where the presence or absence of one species creates, modifies, or destroys the habitat for another species. So 2 examples from elsewhere in the world are woodpeckers. So woodpeckers can promote holes in trees, and those holes can be habitat for other species. Similarly, beavers cut down trees and make these dams in rivers, and they slow up the water, cause these large sort of dams, and clearly that totally changes the environment, makes a new habitat for other species. A very good example of that in Australia are mammals that dig, and unfortunately quite a few of these species have greatly reduced abundance, and that may have wide-ranging effects on the ecosystem. So this bioturbation, or digging and burrowing, is particularly prominent in some of the bandicoots, especially in the bilbies and the wombats. Not only for foraging but also for burrowing. So some of these things can have a huge amount of soil turnover, so a huge amount of digging. This shows the annual soil turnover in terms of tons per individual per kilogram body mass. So the bilby can turn over 20 tons of soil per year per kilogram body mass. So an enormous amount of digging and burrowing can occur. And this has lots of positive effects on the ecosystem. So these mammal diggings can be spots that allow water to infiltrate more easily into the soil. These little holes can trap leaf litter and seeds and other organic matter. And they become sort of areas of increased microbial activity with breakdown of this organic matter in this more moist environment. And it's really important for soil nutrient cycling in these often quite nutrient poor environments, and for recruitment of seedlings. So little seeds can get caught in there. It's a little sort of microclimate, can allow regeneration of environments where you have these mammal digging. So a lot of these bilbies and wombats and other digging bandicoots have become less abundant, and these small rodents as well that dig burrows. And then the soil becomes more harder, it's less permeable to water. You get less microbe activity. There's no safe sites for seeds. It becomes harder for seeds to sort of take off, and you get reduced recruitment and plant growth. So it has some the lack of these vertebrate mammals that do all this digging can cause these negative effects that have wide-ranging implications for the ecosystem. Yes, like I said, a lot of these animals, these mammals that do digging for foraging or for nesting have become threatened or even extinct because of what we've done to what Europeans have done to the environment the last 200 years or so. So this blue colour shows the percentage of the land mass that they have occurred on historically, in the light blue versus their current distribution. So fortunately, the short-beaked echidna is still doing very well, still common throughout the whole of Australia pretty much. In stark contrast to many of the bandicoots, the numbat and the bilbies, booty and the woylie, there's small macropods, potoroos basically. Their distributions have been reduced extensively. So where they did serve this important function of maybe say for the pig-footed bandicoot, the golden bandicoot, there's something like 50% of the land mass, now they're not there at all. So these environments are no longer being dug up in the way they used to be. And that's where I want to end it at the moment. So in this lecture we covered the following topics: phylogenetics. We looked at cladograms, how they're made based on derived characters, and we looked at some definitions. Then we looked at the defining characteristics of phylum Chordata. We looked at the subphylum Vertebrata, its evolution and the derived characteristics of vertebrates. Then we looked at the jawless vertebrates, the extinct and the living forms, and then moved on to the gnathostomes. We looked at how jaws have evolved and the derived characteristics of jawed vertebrates. And then we looked at a little bit about the biodiversity of vertebrates. We looked at global patterns in vertebrate biodiversity, zoogeographic realms and latitudinal gradients, and we looked at two major functions of vertebrate biodiversity, trophic cascades and ecosystem engineers. And that's the end of the lecture for week 1. Please back this up by reading the textbook, the chapters in the textbook, and also I put in the Views site a paper by Klaus Roeder, I remember now, about latitudinal gradients and species diversity. You can read that if you like. I think it's very interesting. And I'll see you next week.